This is Daisy. Since Daisy is a sexually reproducing organism, her cells undergo a special process called meiosis. During this time, it is her body's goal to create gametes. Although the process may seem complicated, it's actually pretty simple. Let's check it out. Before we begin, it's important to note that dogs actually have 78 chromosomes and give 39 to each gamete. However, for our purposes, we're going to use 4. During interphase of meiosis, the majority of cell growth occurs and the cell synthesizes new proteins. DNA is also replicated, and many organelles and proteins needed for divisions are made, such as the copying of the centrioles, which organize chromosomes during cell division.
During prophase 1, chromosomes become visible as the DNA wraps tightly around histones. Centrioles located outside the nucleus move to opposite sides of the cell. The nucleolus and the nuclear membrane break down.
Likewise, homologous pairs are formed to make a tetrad, which is 2 chromosomes with the same genes, but maybe not the same alleles.
The centrioles release spindle microtubules that grow towards chromosomes.
Also, crossing over occurs, which is when the homologous chromosomes exchange portions of chromatids, producing a new combination of genes.
During metaphase 1, tetrads line up along the middle of the cell and remain in pairs, spindle fibers attached to centromeres.
Anaphase 1 occurs when the homologous chromosomes are pulled to opposite sides of the cell by the spindle fibers.
During cytokinesis and telophase 1, the division of cytoplasm occurs. The cell membrane pinches in and forms a cleavage furrow.
The chromosomes uncoil back to chromatin and disperse. The nuclear membrane and nucleus reform in each developing cell, and spindle microtubules break apart and disappear. At the end, each set of homologous chromosomes have formed 2 genetically different haploid daughter cells.
2 haploid daughter cells undergo meiosis too at the same time. However, for our purposes, we'll be individually focused on one. Those that undergo interphase 2 only perform G1 and G2 phase, because if S phase were to be performed, the DNA would be copied again. During prophase 2, the nuclear membrane and nucleolus break down. Spindle fibers form and the centrioles move to opposite sides of the cell. In metaphase 2, the chromosomes line up along the middle of the cell and the spindles attach to the centromeres. During anaphase 2, the sister chromatids are split and pulled to opposite sides by the spindle fibers. In telophase and cytokinesis 2, the chromosomes will uncoil, as well as the nuclear membrane and nucleolus reforming. The spindles will break down and a cleavage furrow forms between the 2 daughter cells. The final product of female meiosis is an ovum, which is created due to the uneven division of cytoplasm, and therefore Daisy is ready to reproduce.